grades, our teacher might have asked us to germinate seeds on wet cotton. What changes do we notice when a seed germinates? We see that at the beginning of the germination process, the seed swells, absorbing a high quantity of water. Later, the seed coat breaks and plumule and radicle emerges out of the seed. The plumule give rise to a shoot system and the radicle give rise to a root system. How does this change take place? What makes them give rise to shoot and root? Let's learn about them in detail. We know that meristems are the tissues that helps to add new cell into the plant body. Shoot apical meristem adds cells in the shoot, whereas root apical meristem adds cells in the root. But what happens to the new cells formed by the meristematic tissues? Will these cells also form new cells? The answer is no. The cells lose their dividing capacity and mature to perform a specific function. Do you know what we call this process as? This process is known as differentiation. During differentiation, the cell undergoes many structural changes and forms permanent tissues. To make the structure and function more clear, let's take an example of a parenchyma cell. The cell has a large vacuole. Nucleus is pushed towards the periphery, and most importantly, the cell has to perform functions such as photosynthesis or storage. Similarly, try to think of more such examples. We have just mentioned that when a cell becomes permanent, it loses its dividing capacity. But is it possible for a permanent cell to regain the capacity of division? Plant shows a variety of interesting phenomena. Dedifferentiation is one such phenomena where a cell regains the capacity of division under certain conditions. For example, the interfascicular cambium and cord cambium divides to form new cells during secondary growth. Now, what happens to the cell formed by the activity of interfascicular and cord cambium? Yes, these cells again lose their capacity of division and mature to perform specific function. This process is known as redifferentiation. Can you list some plant tissues that are formed due to redifferentiation? We know that when a cell gets cancerous, it divides unconditionally and forms tumors. Right? How can we describe tumor in terms of differentiation? Tumor is formed by the process of dedifferentiation when a mature cell regains the capacity of cell division. The same process of dedifferentiation is seen in parenchymatous cells that is used for tissue culture. In the last session, we saw that the growth in plant is open. That is, it can be determinate or indeterminate. On the same note, we say that differentiation in plant is open. But why is it so? Now just think, when a meristematic cell is getting mature and loses its dividing capacity, the type of cell it will be at its maturity is determined on where the cell will be located. For example, if the cell is to be towards the periphery of the stem, it will become epidermis. If the cell is at the center, it will become parenchyma. Hence, differentiation is determined by the position of the cell in plants. Since we have already studied about growth, let me ask you one thing. What is the major difference between growth and development? Exactly. Growth is an irreversible change in the size and shape of an organism, whereas development includes all changes that an organism goes through during its life cycle, from germination of the seed to senescence. Let's look at this illustration to understand development in a better way. In this illustration, you can see that when a meristematic cell undergoes plasmatic growth, expansion, maturation and differentiation, it will form mature cell. Whereas if it undergoes only plasmatic growth, it will divide again to form a meristematic cell. Similarly, a mature cell loses the capacity of division. It matures and ultimately it dies. Have you ever heard of the term plasticity? In many plants such as cotton, coriander, etc., heterophyly can be seen. It means that a single plant has a difference in the shape of leaves, and the leaves of the juvenile plant are different from those of the matured plant. If we observe the buttercups, the leaves produced above water and the leaves produced in water shows heterophyly. The cited examples are of plasticity. Now, can you say what plasticity is? The ability of plant to follow different pathways in response to the environment or phases of life to form different kinds of structure is known as plasticity. So, in short, we can say that development is a sum total of growth and differentiation. Now, what are the factors that control development? Development in plants is controlled by both external and internal factors. The internal factors include plant growth regulators, and the external factors such as light, temperature, water, oxygen, nutrition, etc. have already been discussed. We learned about plasticity in this session. Find out how plasticity becomes helpful to plants. Let's summarize what we have learned.
Manorama Horizon. Learning made simple.